Hello everybody and welcome back to Biomolecules. So in the previous lecture we have started with the sequencing of proteins or peptides, and we have seen two sequencing methods. One is the Sanger method, Sanger peptide sequencing method. And this method is usually used to determine the N-terminal amino acid of the peptide or the protein sample. For N-terminal amino acid. So, mostly what is the amino acid present in the N-terminal of the protein, that to determine that Sanger method is used. Of course, we have seen that this is a chemical method, and you usually use this compound, say aromatic compound with a fluoride here and 2 nitro group. This is the reagent that is used for Sanger's method initially. Nowadays this has been or this can be replaced with the fluorophores or fluorescence containing molecules, such as I have shown the structure in the previous class: diacetyl chloride or diacetyl chloride. So that was the one of the first method. The Sanger's method was one of the first method to determine the sequence of proteins, but ideally we use it to determine only the N-terminal amino acid. Next, we have seen in order to determine the full sequence of the peptide or the full sequence of the protein, we have used Edman degradation method. Edman degradation, and as we have seen that this is also a destructive method or destructive protocol, because you do not get back your sample. And this is the idea of Edman's degradation method, is to cleave the peptide bonds in a sequential manner, one after the other. So, sequential degradation of the peptide bonds. Or cleavage of the peptide bonds that are present in your protein sample. And the reagent that we have used is also a chemical reagent, that was isocyanate, phenyl isothiocyanate. So nowadays advanced degradation method is used, it is pre-programmed, and there is just like PCR machine, there is also the peptide sequencer, like you have DNA synthesizer or DNA sequencer. Similarly, to sequence a peptide, nowadays the machines are there which are pre-programmed, and so it basically cleaves the peptide bonds one after the other, and the cleaved part is eluted out every time and is getting determined by some characterization techniques, usually with the mass spectrometry. So, very briefly we have seen how the advanced degradation method worked. So, if you have a peptide at the N-terminal end, 1, 2, 3, 4, So these are the peptide bonds. Here it is a peptide bond, this is another peptide bond, this is another peptide bond, and so on. So first thing is that the free amine at the N-terminal end that reacts with the phenyl isothiocyanate, and we have given it a symbol as this. So you basically form this kind of complex N. You can write the N, or I am not writing the N directly, this. So you get a marker of this reacting with the amine group. So once this is done, then comes the degradation. It breaks this peptide bond once this is formed by sequential reaction. So this is basically a carboxyl terminal, plus you cleave this peptide bond, and that releases the free amine at the second amino acid. So this is the first amino acid eluted out, and then the entire peptide sequence is left over with an exposure of another amine. Now this amine further reacts with your phenyl isothiocyanate. So you get the next marker, this is eluted out, and it can be determined immediately. So that will tell you the identity of the amino acid 1. So you can know what is the first amino acid. Similarly, once this is done, then comes the cleavage of this bond at the C terminal end. The cleavage of the bond which is present, of the peptide bond which is present after the amino acid which is tagged. So after 2, between 2 and 3, this bond would be cleaved, and it will leave you with 2 with the free carboxyl that would be eluted out. So this is out, plus the rest of the peptide or the protein starting with 3, 4 and the other, with again exposure of another free amine, and this will go on and on. So by determining or by characterizing each of these fragments that are coming out every time, you can know the entire sequence of the protein. That was the advanced degradation method. So ideally these 3 are the methods that are used for determining the sequence of a given protein sample. One is the Sanger's method, mostly used only for the N-terminal nowadays. Second is and most widely used is the advanced degradation method, which is nowadays done automatically in an automated machine. The third one is a technique, it is a instrumental technique known as mass spectrometry. So using high-end mass spectrometric analysis, nowadays you can find out about the sequencing of the peptide. So later on when we will be on the module of learning the modern techniques, I might talk about this mass spectrometric technique. So these are the 3 methods, that 3 ways that are adapted to determine the protein sequencing. Now all these techniques, be it the Sanger method, be it the advanced degradation method, or be it the pure mass spectrometry, all these methods are usually work better or are usually applicable only for short peptides. If you use a very long protein sequence which has 500 amino acids, for example, very long protein sequences, that cannot be used as a sample for all these methods. So that is the limitation of the protein sequencing methods. So large protein sequences cannot be directly used as sample for protein or peptide sequencing methods. So, that is the limitation of these techniques. Large proteins, original proteins that you have isolated from some source cannot be used directly to determine its amino acid sequence. What these methods are usually for, Sanger's method, Edmonds degradation method or the mass spectrometric method, they usually use shorter peptide sequences. So, sequencing methods, peptide sequencing methods I will write now. Peptide sequencing methods are useful for shorter peptides such as maybe 40 to 60, 70, let us say 60 amino acids long. So, 300, 400, 500 amino acids long protein sequences cannot be determined. Short sequences of peptides are the best as a sample in these methods. You can use these short sequences to find out their exact amino acid chain length. So, what is the way out? How can you then, if you cannot use the full protein as the directly in these methods, using these methods, if you are not able to determine the sequence of the full protein, then what is the way out to understand the sequence of this full protein? The idea is, what you have to do now is, if you have a protein, full length protein, that you have to break down into short pieces of the peptides, which will have in this length maybe 40 to 60 amino acids length. So, protein first degraded into shorter lengths of peptides. So, you take your protein sample, treat it in some way so that produces a number of short peptides. Each of them maybe have each of them would have the length below 40 to 60 amino acids, and then all those fragments, all those short lengths of peptides that you have made out of this protein can be used separately to find out the sequence. After this, you can use the advanced degradation method to determine the sequence of these shorter fragments. So, in this case, you have to break down first the large proteins to shorter peptides. That is very important. Now, question is how to do that. So, I will show you two methods. There are actually two ways of doing that. One is the enzymatic method, second is the chemical method. So, first let us take, so there is enzymatic method, and the second is the chemical way to break down a protein into pieces. Enzymatic method. So, you have to use an enzyme which can cleave a protein into pieces. Not only that, it has to be specific. The enzyme has to be such that it will cleave the protein at a specific amino acids. Then only you can know where the cleavage is occurring. It cannot be arbitrary cleavage. It has to be predetermined cleavage, so that you know where my bonds have been broken, and then you can take the actions accordingly. So, such kinds of enzymes which cleaves the proteins are known as proteolytic enzymes. Proteolytic enzymes, or they are also known as peptides. Peptidase means that cleaves a peptide bond. There are many examples of such peptidase or such proteolytic enzymes that are present in our body, that are present in other animals, that are present in plants, and they have very important applications actually. Especially the proteolytic enzymes or the peptidase are very important in our metabolic system. When we eat some food, protein rich food for example, then because the protein is so long, it is difficult to get digested. So, these peptidases or these proteolytic enzymes would cleave your food or cleave the proteins present in the food into short sequences, into pieces, and then your digestion or the metabolism becomes easier. So, these kinds of enzymes are present everywhere. I will give you some examples. So, papain or papain is one such enzyme that is present in papaya, and this enzyme cleaves the proteins into short sequences. So, I do not know, it is a school chemistry. If you have noticed that when you cook food, some especially the protein rich food, for example meat, if you add papaya to it, then it takes lesser time to boil, or it takes lesser time to cook the food. Your meat gets softened very easily if you add papaya there. So, the reason is this, because you get the enzyme papain, which is a proteolytic enzyme, and that cleaves the protein in the meat into the smaller fragments, and then your cooking becomes easier. So, other kinds of examples of the proteolytic enzymes are, which we will be using now, is trypsin or chymotrypsin. So, this is one proteolytic enzyme, this is another proteolytic enzymes. Trypsin and chymotrypsins are usually that you find in the market are usually isolated from pigs. So, how are they specific? How are they specific to sequences? Where do they cleave in the protein structure? If you use trypsin, it actually cleaves the peptide bond, because it is a peptide, so it cleaves the peptide bond. It cleaves the peptide bond at the C terminal position, C terminal of the amino acid either lysine or arginine. So, if you have I am starting somewhere at the middle. This is the N terminal NH. This is your R1. R2. If you have a lysine here or an arginine here, then comes CO, NH, R1, R2, so this is R3, so I will write R4 here, and so on, it goes on. So this is the C terminal, this is the N terminal. If you treat this with trypsin, then it cleaves at the C terminal of lysine or arginine. That means here, this peptide bond, this is the peptide bond after lysine. After lysine, this is the carbonyl group. So this is the C terminal part of the lysine. So after lysine, the following peptide bond would be selectively cleaved by the enzyme trypsin. It can be either lysine or it can be arginine or both. So wherever there is lysine, wherever there is arginine, the following peptide bond would be chopped off by trypsin. So what you will get? You will find amine, R1, CO, NH, R2, CO, NH, lysine, this is cleaved. So you have a free carboxylic acid here, plus your protein is now fragmented into two pieces at least. One is this here, other one is this. Amine, R4, CO, and the other part, the rest of it. So this would be one fragment, this would be another fragment. So that's how trypsin would cleave at a specific amino acid sequence in the protein. So if you take a whole protein and if you treat it with trypsin, then you can know where your trypsin is cleaving, which amino acids or which peptide bonds this is cleaving. And the fragments that you get after the reaction, that you can isolate each one of them, you can separate out, and each of them you can put it or you can use further for advanced degradation method. So once you have the fragments, next step is you purify each fragment and then use it for advanced degradation. So in this way you will know the sequence, exact sequence of each fragment, and then you can basically tie them up to get the full sequence of the protein. So that is the mechanism of action of trypsin. Now if you have chymotrypsin, chymotrypsin is another proteolytic enzyme or another peptidase, and it cleaves the peptide bond at the C terminal end of tryptophan. TRP. So if you have a protein sequence, if you treat it with chymotrypsin, wherever there is tryptophan, it will cleave the following peptide bonds and you will get the shorter fragments of peptides. Again, you can purify each of these fragments and then use them for advanced degradation method. So, that is how whenever you will be using an enzyme, it has to, you have to select an enzyme for which you know where it cleaves. That is very important actually. Predetermined sequence have to be cleaved. Now comes the chemical method. In chemical method, you use a reagent, of course, and this reagent is CNBr. This is cyanogen bromide. This is the reagent that is used to cleave a peptide bond at a predetermined amino acid. So, this again cleaves the peptide bond again at the C terminal end or C terminal position of a specific amino acid, in this case is methionine. Met. So, CNBr cleaves all the peptide bonds wherever there is a methionine. How does it work? If you have, so this is the N terminal, there can be other peptide, other sequence. Here this is R1, CO, NH. Let us say here you have a methionine, CH2, CH2, methionine is S and CH3. This is methionine. This is the C terminal end. So, this is the C terminal peptide sequence R1, R2, this can be R3 and it goes like this. So, it should be CO and then the rest of it. Let us take that this is the sequence of the protein, and if you treat it with CNBr, then what happens? Bromide is a good leaving group, it is electronegative group, so it will try to drag the electron density and move away. Sulfur has lone pair of electrons and sulfur has a large size. So, it has the lone pair of electrons is highly donatable because it is to some extent electropositive. So, the lone pair of electrons will attack the carbon of the cyanide, eliminating the bromine or the bromide. So, you will have CO, NH, here CH2, CH2, S, CH3 and cyanide, and this would be now plus because it has donated the electron. So, sulfur plus NH, R3 and this. Now, this has formed the positive charge here and it has this. Now, if you look at cyanide, that is why cyanide has been used. Cyanide or Cn minus has dual character. So, in this case in cyanide also is pretty stable as cyanide minus. So, it has the tendency also to drag the electron density towards itself, because nitrogen is electronegative. So, the electron density would be dragged towards the nitrogen, that forces the electron density of this bond to be dragged towards the cyanide. That makes the sulfur even more electropositive, even more electron deficient. It has already a plus charge, and because of the cyanide attraction, electron withdrawing effect, the positive charge would be, or positive charge density would be higher here. So, this whole thing becomes a very good leaving group. Since this is highly electron deficient, it will try to take away the bonded electron and move away. So, it is very easy to cleave this bond. So, if you hydrolyze it in presence of water, the carbonyl here, that is where the chemistry of the peptide bond is coming. This, although it is not very reactive, peptide bond is not very reactive, but because you have a pretty good leaving group, this reaction happens. It will cleave, it will attack this carbon, eliminating the whole thing, the sulfide, and you get a cyclic compound which will look like this. Is CH3 CN, this is positive charge, that makes it a good leaving group. CONH, here this is R1, R2, this is R3. So, that will attack this carbon and this would leave. So, what you have is, of course, this is with treatment with water. R1 CO NH, this O here. So, this bonded electron would go, and then this would be the resonance. This is gone, this is coming here, this is attacking. This is the carbon, and this would be your NH. This is your R3 CO and the rest of it. Here you have formed the double bond and it would be NH plus. So, this is the cyclized compound that would be forming through the oxygen, and to stabilize this there will be this resonating structure. Now, this becomes a double bond with the positive charge on NH amine, or with the positive charge on nitrogen, which is also not very stable. So, it will undergo a rapid hydrolysis. Rapid hydrolysis, and this bond would be cleaved. If you hydrolyze a carbon nitrogen bond, what you will have? Water will attack here, you will move this. So, you will basically get a carbonyl compound here. CO NH this, here you will have a carbonyl, plus you will have the free amine NH2 R3 this. So, you have a breaking or the cleavage of the peptide bond in the position where there was a methionine, or the following peptide bond after the methionine. So, this is the chemistry here, and now you can again separate the individual fragments and do the advanced degradation. So, the number of fragments will tell you. So, if you have 5 fragments, for example, in the whole protein sequence, then you know you have at least 5 methionine present in this at the terminal of the wherever there was the cleavage. Okay. So, that is how you can actually break a given protein into its shorter peptide sequences. So, now I will give you two problems. Problem 1 is, let us consider a protein sequence. N-terminal, glycine, aspartic acid, phenylalanine, lysine, tryptophan, arginine, phenylalanine, alanine, methionine, and this is your C terminal. Let us say this is a given protein sequence. Question A is, write the fragments if treated with trypsin. Question B, write the fragments if treated with chymotrypsin. So, can you write down the fragment of the proteins, or what are the fragments that you will obtain if you treat this given sequence with trypsin? What will happen if you treat it with trypsin? It will cleave the peptide bond at the C terminal position of wherever there is a lysine or wherever there is an arginine. So, glycine, aspartic acid, phenylalanine, lysine, tryptophan, arginine, phenylalanine, alanine, methionine. So, if you treat this with trypsin, then it will cleave here, there is lysine here. So, after the lysine you have another arginine here to cleave also here. So, the fragments that we will see is glycine, aspartic acid, phenylalanine, lysine, acid, free acid plus NH2, tryptophan, arginine, free acid plus it will expose the amine at the phenylalanine, methionine. So, this is the C terminal end. So, this is the C terminal. So, these are the three fragments that you can expect if you treat this specific protein sequence with trypsin. Now, B is glycine, aspartic acid, phenylalanine, lysine, tryptophan, arginine, phenylalanine, alanine, methionine, and you are treating this with chymotrypsin. Chymotrypsin, and we know that chymotrypsin cleaves the tryptophan, the C terminal end of the tryptophan. So, it will basically cleave here. cleaves after tryptophan, and there is only, I think, one tryptophan. So, you will have a single cleavage here, and your fragments would be glycine, aspartic acid, phenylalanine, lysine, tryptophan. This should be chopped off. It will expose free carboxylic acid plus free amine of the next arginine and the rest of it, alanine, methionine. So, these are the two fragments that you can expect from here. So, I will give you another question. Let us take a different sequence: alanine, glutamic acid, arginine, valine, leucine, methionine, phenylalanine, TRP tryptophan, alanine. So, this is the given sequence. Question A is, if you treat this, treat with CNBr, then what are the fragments? CNBr followed by, of course, this is very important, followed by hydrolysis. Treatment of this sequence with CNBr followed by hydrolysis. What are the fragments that you will find? CNBr would break after methionine, right? So, here this should be broken, and you will end up with alanine, glutamic acid, arginine, valine, leucine, methionine. Of course, no free carboxylic acid. It will finish here with the cyclization. So, I am writing this as cyclic. You do not have to write anything actually. So, it is methionine plus it will expose the amine of the next amino acid, which is phenylalanine, tryptophan, alanine, and that is your C terminal. So, these are the two fragments that you will obtain. Second question is, first treat with trypsin and then with CNBr. So, it is a combination of treatment with enzyme as well as the chemical reagent. You are treating both with trypsin and with CNBr. Then what are the fragments that will that you can expect? This, of course, with the expectation that they will not interfere into each other's business. So, if you treat it with trypsin, what you will have? You will have a cleavage after arginine. You will have only arginine here. So, let us write that: alanine, glutamine, arginine COOH plus NH2 valine, leucine. Then you have methionine. You have used CNBr, so it will cleave after methionine. It will stop there, plus amine, phenylalanine, tryptophan, alanine. These would be your products. These are the 3 fragments that you will obtain. I guess I was correct. Trypsin, arginine, there is only arginine, no lysine, so only one cleavage here for this, and another for the methionine, 2%. 2 cleavage, that will give you 3 components. So 3 fragments that you can expect by a combinatorial treatment of trypsin and cyanobromide or cyanogen bromide, this plus this plus this. So likewise, you can actually find out, you can play around with the different sequences. So if you take a large protein sequence and treat it with a combination of trypsin and cyanobromide, or even a combination of trypsin plus chymotrypsin plus cyanobromide, then what are the fragments? You can expect a huge number of fragments that will be present in your final products, and you can separate each one of them. You can expect that your protein is now degraded enough into the short sequences which you can use for advanced degradation method to know its sequences. So this is roughly about the sequencing of the proteins. If you have a protein, unknown protein sequence, how would you find out its the amino acid chain? What are the amino acids present one after the other? We have talked about 3 methods for the sequencing itself, and then today mostly we have talked about the methods through which you can actually cleave, or you can actually make fragments from a large protein into its smaller pieces. Okay. That is for today, and then we will take the next lecture and we will talk about the peptide synthesis, solid phase peptide synthesis that we will be using in the laboratory method to construct a peptide sequence. Thank you.